We've actually been doing cognitive neuroscience for a long time, hundreds and thousands of years, in fact, because in a very primitive way, though, but because we've learned a lot about the brain from the examinations of people that have suffered head traumas and brain injuries. So whether it was, Flavius Maximus getting his, skull cracked open in a battle, or a gladiator getting his, a spear through his head in the arena, or somebody in the Middle Ages having a stroke after they were bled. We've been able to tie together a lot of information about cognitive deficits related to cognitive abilities. And we've been able to see the relationship between brain function and cognitive abilities. Lately, though, we have much more sophisticated methods, brain imaging, so we can look inside your head without killing you, and other different ways to measure brain function through in live patients. Another thing that we've done quite a bit is through clinic. We've looked at clinical case studies. One of the most famous clinical case studies is the case of poor Phineas Gage. So poor Phineas, I say poor Phineas because Phineas, he was a nice guy. He was working as a laborer on a construction crew, on a railroad construction crew in the 1800s in Great Britain, in England. And he was in the process of packing in some dynamite blasting caps into a hole that had been drilled in a rock. And he was using a long metal steel rod to pack these blasting caps into this hole drilled into this rock. And as you know, when you rub rock and steel together, you create a spark, and that's exactly what he did. He created a spark which set off the blasting caps that were in the hole. The rod shot out of the hole, hit him in the cheek beneath his eye, and popped out through the top of his head. He essentially gave himself a frontal lobotomy. And poor Phineas, after so he survived this railroad accident, and was damaged in the frontal part of his brain severely. He ended up not able to work anymore, but he did make a little bit of money working as a freak in circus shows, believe it or not. But one of the things that happened to him was that he was less able to control his emotions, and his decision-making abilities were completely suffered a great deal. And we learned, based on that case that was documented by many doctors that examined Phineas, we learned that the frontal lobe, that part of the part of your brain that's right there behind your forehead, is important for helping us to regulate emotion as well as decision making. Particularly the prefrontal cortex, the very front part of that cortex, is critical in helping us to make decisions. Another clinical patient that we've looked at is a guy who has been identified as Tan, T-A-N. Tan wasn't able to speak for many years, and after he passed away, his brain was studied. And Paul Broca found that he had damage to his left frontal lobe. So the left part of his frontal lobe near the front of the temporal lobe. So kind of in the frontish area there of the frontal lobe, right there, kind of like right between the frontal and parietal lobes. And it was a very specific lesion that this guy had right there on that little spot. And ultimately, this area came to be named Broca's area, after Paul Broca, the guy who discovered it. And it turns out that anybody who has damage in Broca's area creates a disorder where a person has difficulty producing speech, so they can understand what's being said to them, but they have trouble producing speech, generating speech. So if you've ever had a relative or a friend who's had a stroke and they have a lot of trouble talking, you've seen that. One of the areas that was damaged would have been Broca's area. Carl Wernicke discovered Wernicke's area, which is located again in the left temporal lobe, so it's further back in the temporal lobe, close to the front of the occipital lobe. And it turns out that damage to Wernicke's area causes a deficit in language comprehension and meaningful language production. So people with damage to Wernicke's area are unable to understand spoken words, and they can produce a lot of spoken words, but the words that they produce are not meaningfully connected. So they produce what's called word salad, just this meaningless jumble of words, which is kind of scary. But for both Broca's area and Wernicke, it was the clinical case studies that these guys initiated that allowed us to understand these areas of the brain and brain function. So Oliver Sacks, who's was a very famous neurosurgeon, wrote actually some interesting books on this topic. Excuse me. So Sacks had a patient who had difficulty distinguishing between living and non-living objects. So this person would, for example, have trouble distinguishing between, say, a cat and a cup. Telling which one was alive and which one wasn't. This is known as object agnosia, a big problem with object identification. And typically, this type of damage, this type of agnosia results from damage to the lower temporal cortex. And since the temporal cortex is the temporal cortex is intimately related to language functions, this suggests that the deficit really isn't related so much to the ability to perceive the object. Rather, it's related to language production, so the ability to link the proper words to the object. One of the most famous cases in neurosurgery, in neuroscience, with regard to human brain function and memory, was his patient named HM. He went by the initials HM to anonymize him during the whole research that he was undergoing. His name was released after he passed away, and he just passed away like not even 10 years ago. It was fairly recently. Henry Molaison, I can never pronounce his name, but anyway, poor Henry, when he was a young man, he actually had severe epilepsy, temporal lobe epilepsy. It was this epilepsy that would originate in his temporal lobe and spread throughout his brain and cause severe seizures. So in a last-ditch effort to relieve Henry of his seizures, they did brain surgery and removed his temporal lobes. Back then, they didn't really understand what the different parts of the brain did, and in the process of sucking out his temporal lobes from his head, they also removed a large part of the brain called the hippocampus, which is a large structure underneath the cortex, and a lot of it curls into the temporal lobe of the brain. Anyway, HM ended up getting not only his temporal lobe removed, but also his hippocampus. And after the surgery, Henry, was quite unable to learn anything new. He had some memory of events prior to the surgery, but from the point of surgery on, after he recovered, he was unable to learn anything new. So if you were a person he had never met before, You'd go in and introduce yourself, and then, within a minute or two, he would forget that he ever even saw you. Which is a horrible thing to have happen, especially to, who apparently was a very nice guy like poor Henry. But he was unable to form new memories, so he was intensively studied, and from his case, we learned of the importance of the hippocampus and memory abilities. And we also learned that the hippocampus is not necessary for forming and retrieving all types of memories, because like I said, Henry could remember some things, several things from his past. He just couldn't remember everything, but a lot he could remember. And but he was unable to learn anything new, which it turns out that was not quite true, because one of the weird things about Henry's condition was that one of the things they would do is give him puzzles to play with. So he would actually, not to play with, but to test him. So they give him this puzzle that he had to put together. And of course, if you asked him, have you seen this puzzle before? Have you ever put this puzzle before together? He'd say, well, no, I've never seen it before. However, they would give him the same puzzle, the exact same puzzle, over and over again. And of course, he would claim that he had never seen it before. But interestingly enough, for some of those puzzles, he got better at it. He would actually get better and better and put them together faster and faster. So even though he was not aware of the fact that he had seen it before, or that he was even getting better at it, there was some motor type memory in there that was still being encoded and stored, helping him to get better at putting those puzzles together. One of the advantages of doing clinical case studies is that it provides us clues to areas of the brain that are most important for different types of cognitive tasks, right? So it lets us link together what parts of the brain might be important for different tasks. Now, one of the things you need to understand about brain function and brain research is the fact that we don't have little melon balls of function in our head, right? So there's not like a memory area. There's not a area that helps you recognize your cat. There's not an area for controlling hunger. There's not an area for controlling satiety. There's not an area for controlling thirst. We don't, that's the old melon ball theory of brain function that my graduate school mentor Vern Cox used to talk about all the time. And that, we don't actually have melon balls, but we have discovered that different types of things that we understand as functions and that we understand as behaviors and functions do seem to involve certain areas of the brain. So there is a link between certain areas of the brain and certain types of things that we do. So for example, the hippocampus, it's pretty critically involved in learning and memory. It's involved in a lot of other things too, but it's involved critically in learning and memory. The disadvantage to clinical case studies is that the damage is not controlled by researchers in humans. If you're studying somebody that had a stroke, I mean, the damage is never localized. Very rarely, I should say, localized. I mean, you're going to have damage all across the cortex and sometimes even deeper down. And you can't control it. So any of the results that you get are going to have to be very you have to be very careful about talking about relating that type of damage in that patient to the specific damage that patient has undergone. Of course, in animals, when we do brain lesion studies in animals, we can do chemical lesions, electrolytic lesions. There are all kinds of genetic manipulations that we can do nowadays to turn on and off or deactivate or stimulate different parts of the brain. And we've gotten some really good, many, many years of great data on that. And that has helped us immensely to understand brain function. So let's talk a little bit now about the structure of the nervous system. As you know, the brain is composed of billions of microscopic neurons. And we have gotten to the point now. Where our technology is actually good enough to where we've been able to understand a lot about what neurons do. So we can record neurons, individual neurons. We can, we even have genetic techniques now where we can insert genes into neurons. It's called optogenetics, where we can insert genes into neurons that will allow those neurons to be turned on or off whenever you shine a little tiny light inside that part of the brain. So it's a really neat way to create these mice or rats that have these optogenetically enhanced brain parts, and then you insert a little light fiber into their brain, and whenever you want to turn that part of the brain on or off, you just shine a light, and the protein changes its shape, and you essentially create like a temporary lesion or a temporary activation of a certain part of the brain. It's a really neat way to study the brain. So how all of this studying of the brain, to understand the study of the brain, you have to understand some of the relevant structure and function of the brain. So let's go through that real quick. As I hope you know, the brain is composed of billions of microscopic cells called neurons. Neurons are the basic cells of the brain. Neurons are the cells of the brain that communicate with each other. They are networked together, and they can talk to each other, and in this way create vast, complex networks that we believe are used to analyze inputs and produce all of the behaviors and thoughts and feelings that we experience. Neurons are enormously complex. We study them and we get more and more detailed. And every time we go down to a level of detail in our studies of neurons, they just become more complex. We just open up another, dimension of complexity. Neurons communicate via the production and excretion of chemicals called neurotransmitters that live in the very tips at the ends of the neuron. These chemicals, these neurotransmitters, are squirted out of the tips of the neuron whenever the neuron is stimulated, or whenever the neuron fires a stimulation down its cable to the tips of the neuron. And the neurotransmitter chemical is excreted out of one end of the neuron, and that neurotransmitter travels across a gap to then it attaches to molecules on the other neuron next door. And in that way, neurons are able to signal each other by the excretion of different neurotransmitters, these chemical signals that are used to for one neuron to signal another neuron. In general, neurons can be broken up into different parts. If you look at a picture of a typical neuron, you will see that there's these tree-like projections that are coming out from the center body, the soma of the neuron. And these tree-like projections are called dendrites. The dendrites are you can think of those as the receiving antennas of the neuron. The dendrites, parts of the dendrites are covered with molecules that are sensitive to neurotransmitters. So the dendrites are sitting there like little trees, little branched trees in three-dimensional space, and connected to those dendrites are the tips of other neurons. And whenever a neurotransmitter is released from those tips, the neurotransmitter binds to the chemical receptors That's what they're called, receptors. The molecules are called receptors living on the dendrites. And that causes electrical changes to occur in and around in the area of the dendrites, and those electrical changes are picked up and added together at the axon body. Now you might be wondering what these electrical changes are. I mean, since we're not machines, we're not made of copper and batteries and capacitors and things like that. The electrical activity of a neuron is actually made up of the movement and gradients created by ions. So you can think of a neuron as kind of like an enclosed capsule, right? Inside the capsule, you have lots of negative ions when the neuron is at rest, and the outside, in the extracellular fluid, you have lots of positive ions. And this difference of negative to positive is maintained by pumps that pump the sodium and potassium ions into and out of the cell. And the pumping of these ions it's a continual thing. All of your neurons are doing this. Maintains this electrical charge across the membrane of the neuron. You can actually stick a little wire, tiny little glass wire, into and measure the electrical charge of a particular neuron. And usually you'll get something like around -70 millivolts. So there's an electrical charge due to this differential placement of positive and negative ions inside and outside of the neuron. And at rest, this differential placement of ions causes a potential charge of -70 millivolts. The dendrites receive stimulation from neurotransmitters via their receptors, and that causes little gates to open, little protein gates to open in the membrane that lets in and lets out ions, and it changes the potential a little bit. It goes from -70 millivolts to -69 millivolts to -65 millivolts. And the more stimulation the cell gets, the lower and lower, or the higher and higher goes the it'd be the lower and lower the potential goes. So it goes from -70 millivolts to -65 millivolts. At some point, the neuron is going to get stimulated enough to where that value goes from -70 to -60 to -50 to around -40 or so. And when it hits that value, and it varies from neuron to neuron, but when it hits that value, that's a threshold value. And that causes a big, huge electrical signal to be transmitted down the other part of the neuron known as the axon. This signal is a much larger signal than the little ones that were caused by the changes in the dendrites. It is referred to as an action potential. The action potential is the electrical signal that travels down the cable that comes out of the body of the axon. This cable is referred to as an axon. So the electrical signal bounces down the axon. And essentially what the action potential is, it's the ions rushing in and rushing out of the of different parts of the axon's membrane. So potassium, sodium, and chloride ions are rushing in and out. So sodium flows in, and when the action potential has passed, the potassium flows out, and the sodium channels close up, and the pumps reestablish the -70 millivolts differential for the resting potential. It's kind of like flushing a toilet. Basically, you flush the toilet, and the ions are rushing in and out. And then after you flush the toilet, you have to kind of let it refill right before you can flush it again. That's kind of what's happening here. Is that at rest, the ions are just sitting there on the inside and the outside, maintaining this -70 millivolt resting charge. Whenever an action potential comes down the axon, the protein gates open up, and sodium flows in, and potassium flows out. So that's the flushing of the toilet. And then after sodium, potassium flow in and out, that is the actual action potential. That's the actual signal. The gates close up, and then the pumps work to reestablish, to push the sodium back out, the potassium in, and bring the resting potential back down to minus 70 millivolts. That's the way the whole thing works. This action potential makes its way the action potential makes its way down to the terminals of the axon at the very end. Inside the terminals of the axon, we have what are called little bubbles or vesicles. These little vesicles are little bubbles that live at the very tips of the axon terminals, and the little bubbles are filled with neurotransmitter. The neurotransmitter inside the little bubbles is sealed inside the bubble, and whenever the action potential makes its way down to the axon terminal, those little bubbles move forward. They move forward and connect to the surface, the membrane surface, and then they pop. They release their neurotransmitter into the synaptic gap, which is the area between the dendrite of one neuron and the terminal of another neuron. The neurotransmitter travels across the gap, binds to the receptor on the next door neighbor dendrite, and the whole process repeats itself. There are many different neurotransmitters. Some of them you may have heard of, like dopamine, serotonin, norepinephrine, acetylcholine, GABA. There's and some of these neurotransmitters can either be excitatory, meaning they're more likely to cause the cell to fire an action potential, or they might be inhibitory, which means they might be more likely to cause the cell to not fire an action potential. So it's all a very complex network, controlled by the flow of ions, controlled by the flow of neurotransmitters, and controlled also ultimately by the patterns of firing, the patterns of the way these neurons are all firing and communicating with each other. The human brain, our brain is organized, first of all, we really don't know how the brain works. We know that the brain is organized somehow. It is organized. We're just now trying, really figuring out how it's organized. It's probably organized in networks. And those networks somehow or another underlie specific cognitive functions. We do know that different areas of the brain are specialized for different functions. There is this phenomenon called localization of function. So, for example, there are parts of our cortex that are dedicated to motor behaviors, controlling how we move, and other parts of our cortex that are dedicated to sensation of different parts of our body, parts of our cortex that are dedicated to processing audio, what we hear, parts of our cortex that process visual information, etc., etc. So there is a certain amount of localization of function. There's also lateralization of function, meaning that we have two halves of the brain, the right half and the left half. They're both essentially mirror images of each other. And it turns out that there are certain functions that are on one side of the brain and certain functions that are on the other side of the brain. So, for example, the most generic example is that the right side of your body is controlled by the left half of your brain, and the left half of your body is controlled by the right half of your brain. So if you have a stroke and you're unable to move you're unable to control the movement of your right arm, it means that you had a stroke that affected the motor cortex of your left hemisphere. Please don't get this confused with this stupid notion of right brain, left brain. You guys have heard that before, right brain, left brain. Right brain thinkers are artistic and creative, and left brain thinkers are analytical. There's no evidence for that at all. That all came about probably from the fact that one of the things that is lateralized is language. Language is very strongly lateralized in humans. So if you're a right-handed person, which 90% of us are, your language functions are going to be in the left side of your brain, and vice versa. If you're a lefty, your language functions are on the right side of the brain. We know that many complex cognitive tasks are a function of distributed processing. We know that we know that brain areas work together in systems to process a lot of different types of information. So there's a lot of brain systems that are working together to process pretty much everything that we understand. There is localization of function for cognitive processes, and so there are specific areas for processing words and symbols and recognizing human faces and things of that nature. But for the most part, we know that doing complex cognition involves multiple areas organized together into processing systems. All right, let's talk a little bit about some measures that are used in cognitive neuroscience. Now, a lot of the brain imaging techniques that are used today are actually able to record brain activity in humans as they perform various cognitive tasks. However, not all the techniques rely on the activity of the neuronal cells in the brain. So let's talk about some of these different techniques. So, for example, we have a single cell recording, and this is where basically a very fine recording needle, it's usually a very thin glass needle, is inserted into one neuron in an area of the brain the researcher is interested in. So if you're interested in studying visual information processing, that happens in mammals, that happens in our visual cortex, which is in the back of our brain and the occipital lobe. What you're going to do is you're going to drop an electrode into that part of the brain and find a neuron, and you're going to be able to measure the voltage changes in that neuron as it fires action potentials. You can actually measure the action potentials, and if you connect the measuring device to a little clicker, you can actually, which is what a lot of these labs do, you'll hear the little clicking sound of the action potentials being fired off. Let's see. The single cell recording, it requires surgical insertion. So of course the critter has to be immobile. It's typically used with laboratory animals. One of the discoveries that was made with this technique, it was the discovery of mirror neurons. Mirror neurons are these neurons that seem to fire in response to the motions and movements of others. So that's why it's called mirror, because they are used, we think, to sort of represent what other critters are doing, what's going on with other organisms outside of us. And that's how they were discovered, accidentally, of course, like a lot of these things were discovered accidentally. The Nobel Prize-winning work of Hubel and Wiesel, who discovered visual information processing in the cortex of the cat, was also done with single-cell recording. And here's an example here of a rat that's been put into a stereotaxic instrument, and the electrode has been lowered into this rat's skull. And the rat is anesthetized. It's not alive. It's not like kicking and screaming or anything like that. It's been fully anesthetized. And once the rat's been placed into this device, we can then connect the wires to the electrode that's been lowered into the brain and measure action potentials of any neurons we happen to find. EEGs, electroencephalographs, are used to detect changes in electrical signals of the brain, so gross electrical activity of the brain. Some of you may have had an EEG. Basically, what they do is they put a cap. It's like a cap that's filled with these little flat, round electrodes, and they put it on your head and they strap it down real good on your head. And all of these little electrodes work together to detect the gross electrical activity of the brain. Researchers use EEGs to examine what are called ERPs, or event-related potentials. And basically all an ERP is an event-related potential. All that is, it's just a certain activity that appears in an EEG related to a specific event. So, for example, if I present you with a certain tone, a certain 1,000 or 1 kilohertz tone, I might be able to identify a certain pattern of change in your EEG that only happens whenever you perceive that tone. And I would call that the ERP, the event-related potential for that particular tone. You can look and see the activity of a brain in almost real time. And you can actually determine if there's an effect of a stimulus presentation on neuronal activity. And you can see what general area of the brain that particular that particular activity is happening in. So it's a great way for us to look inside the brain and not have to rely on what people are telling us, what they perceive, what they can perceive. We can also see in a fairly gross way, but we can see what sorts of neuronal activity changes occur as a result of getting a specific stimulus. Magnetoencephalography. I can never say that word. MEG, MEG, involves placing the head of a person in or near an electrical scanner that can detect electrical activity with better location accuracy than an EEG. So this is basically we're putting your head into a big ass magnet that has the magnetism focused in a specific beam. I guess you can think of it as that. And then we can look and see how the magnetic beam is being deflected by the electrical activity in your brain. And in that way, we're able to actually focus the magnetic beam on specific areas of the brain, and therefore get more specific measures of brain activity compared to the EEG. MEG recordings can occur during a task, so that the changes in activity can be detected that correspond to the presentation of cognitive stimuli. So I can, for example, have you I can have you working different types of problems, and I can watch the changes in your brain that happen as you work different types of problems or solve different types of puzzles. MEGs are limited to only recording in the outer cortex. So I can't really record anything that happens, like underneath the cortex. It's really just the outer cortex. Another thing that has been done for, many years actually in animals has been electrical stimulation of neurons. So we've had the ability for many, many years to, lower an electrode, like you saw in that rat before, to lower an electrode into different parts of a rat's brain and deliver a small amount of electrical charge to that electrode and stimulate that part of the rat's brain. And we've learned a lot about brain function using that method. You can also inhibit the firing of neurons temporarily by jamming them with an electrical signal, or even permanently by creating a lesion, a small electrical lesion. In humans, one of the techniques that's used for electrical stimulation is transcranial magnetic stimulation, TMS. And this is essentially where you sit again next to a big ass magnet. And that magnetic field is used to excite or inhibit neuronal activity, to investigate the functioning of different specific areas of the brain. There's also transcranial direct current stimulation, which is a more focused type of transcranial stimulation. It's interesting to note that in some cases of depression, that transcranial magnetic stimulation seems to have an alleviating effect on some people's depression, which I think is kind of cool. We can also look at your brain without killing you, without having to kill you, crack your head open, and pop your brain out. We can actually look at your brain, image your brain while you're doing different things. One of the techniques that's used to do brain imaging is MRI, magnetic resonance imaging. Some of you who have maybe had a back injury or or knee injury or other type of injury have probably had an MRI done. MRIs are basically based on this principle that when you put living tissue into a very strong magnetic field, it causes the hydrogen atoms in our molecules of our body to flip, to reorient their little axis in line with the magnetic field. And we can detect that flip. We can actually detect, when the conduction of electrons through the flipped hydrogen. And it turns out that soft tissues like your guts and your skin and whatnot have different levels of hydrogen concentrations, and bones don't have hardly any hydrogen in them. So it turns out that you can use this differential flipping of hydrogen in different tissues to create a very nice view of different tissues. And we've done a lot with MRIs and using MRIs to relate different brain injuries, for example, to different cognitive deficits. So if you've had a bad stroke, I can get all your symptoms and see what your cognitive deficits are, and then I can look at your MRIs and relate those cognitive deficits specifically to the different areas of the brain that were damaged. Let's see. PET, positron emission tomography, is blood flow detection. We can actually detect how much blood is flowing through different tissues of the brain. Essentially, the way this works is I have you ingest a small amount of a radioactive substance, and that substance, and it's not going to give you cancer or anything like that. It's just enough that can be detected by the sensors on the PET machine. But the PET scan actually goes, and the radioactive substance goes into your blood, and then it ultimately flows to the brain. And the radioactivity in the blood is then measured in different parts of the brain. And it's presumed that if one part of the brain is working harder than another part of the brain, then that part of the brain that's working hard is going to be sucking up more blood. So if it's sucking up more blood, it should glow brighter because it's going to be more radioactive than parts of the brain that aren't working. So by using PET scans, we're able to detect which areas of the brain are linked to the performance of certain cognitive tasks. Now, it's not real time. Some people think that PET scans are real time. It's not real time. So you give people the radioactive stuff, you put them in the PET scanner, you have them do the task, and then you gather the data. Then you have to take that data and analyze it and break it down so that you can reconstruct these images of these of the brain, so that you can see which parts of the brain are active and which parts aren't. fMRI, functional magnetic resonance imaging, is another imaging technique that records brain activity with the scan of the magnetic properties of the blood flowing through the brain. So we're basically looking and measuring the differential. Again, we're measuring the differential flow of blood, but we're not using radioactivity. This time we're just using the properties of the blood itself. And we can again use fMRIs to look at the differential activity of different parts of the brain. Typically, the way this is done is, they'll put you in an MRI machine in one of these machines, and then they'll say, they'll have you like just chill out and relax for a while. They'll get the baseline activity of your brain, and then they'll have you do a task, whether it's reading or listening to music or whatever. And then after that, they'll compare the two, and they'll do like a subtraction method and show that, the difference between doing the task and not doing the task was this. When we're looking at living brain, right, we can basically record the electrical activity of neurons with, with your with your EEGs, right? Or we can do these imaging techniques, right? The advantage to these methods is that they allow cognitive neuroscientists to gain important knowledge about the connection between brain function and cognitive processes, right? So we're able to do that. The disadvantage is that not all cognitive tasks are easily adapted to the brain recording techniques. Not for the most part, these techniques are really good at detecting the activity in the outer cortex. But, there's a lot of things that we do that involve more than just our outer cortex. Lots of different visual information processing, memory, muscle learning, things like that involve vast parts of the brain. So it's kind of hard to get to those. The other disadvantage to these techniques, particularly the brain imaging techniques, is that the machines that we use for them are very expensive. They're expensive and they're hard to get. So, you really have to have a lot of money or plan carefully to do this type of research. Finally, we have the question. We're assuming, right? We're assuming that all mental processes, everything, can be explained in terms of brain activity. So essentially, we're assuming that everything that you're experiencing right now, all of the thoughts, feelings, your behaviors, your motivation, your attention, everything can be explained simply in terms of brain activity. That we can somehow link, this particular phenomenon of behavior or mental process to this particular activity of the brain. We're assuming that, but that's an assumption. We're not really 100% sure that that's even possible. We don't even know that all mental processes can be explained in terms of brain activity. But however, having said that, all of the evidence we have, especially the evidence from imaging and EEG studies, tells us that, yeah, probably. Most everything we do, everything we think, feel, and say, and do, and believe can ultimately be linked back to some type of brain activity.